Good morning, ladies and gentlemen. It's my pleasure to start this session. It's not an ordinary keynote lecture because this lecture is called Gjønnes Medal Lecture since Commission on Electron Crystallography provides every 3 years, on every IUCR Congress a medal to people who contributed the field of electron crystallography the most. Criteria for a selection of a person whom this award will be given each year is based on whether the research opened a new field and how big was its impact and influence to the crystallography community. So this time we had a subcommittee of 5 scientists from Japan, China, USA, and UK, and after thorough consideration, we decided to award the Gjønnes Medal on this IUCR Congress jointly to Professor Richard Henderson and Professor Nigel Unwin for their development and powerful application of structural determination methods for biological complexes and near atomic resolution using electron crystallography. So I would like to ask Professor Henderson and Professor Unwin to come here so we could give you the medal.
Please open your eyes. Both.
So, as you understood, we are going to have 2 lectures, and first will be Professor Nigel Unwin. And he has done his PhD in Cambridge, and from '68 he became staff scientist at the Laboratory of Molecular Biology. From '80 he became professor of cell biology at Stanford University and returned to the Laboratory of Molecular Biology in '88. At '92 he became head of Neurobiology Division, and from 2003 to 2008 joined head of Neurobiology Division. Well, he is fellow of many societies, for example, Royal Society, and was awarded many awards, such as Gregory Aminoff Revolt and Royal Society Krunian Lecture and Medal. So I would like to welcome Professor Nigel Unwin to give his lecture.
Thank you, Louisa. I'd like to thank the Gjønnes subcommittee that selected me and Richard for actually choosing us. And I think it's a really great honor to be framed together with the earlier recipients who are very distinguished scientists. So thank you very, very much. I want to thank the organizers for this wonderful meeting, and I particularly wanted to thank Samar Hasnain for supporting the cryo-EM and also encouraging me to write a paper that actually complements the last part of my talk, so I can keep that bit really brief. So I began my career with electrons actually about 50 years ago, working as a metallurgist And this was as a PhD student in the Cambridge University Metallurgy Department. And what I was trying to do was to look at the grain boundary precipitates between metal grains in an aluminium zinc magnesium alloy, and trying to see how they related to the orientations of the bounding crystals on either side. And you could tell the orientations very accurately from details of the electron diffraction patterns. And what I found was that when you had orientations where you could draw a superlattice between the two bounding crystals, the precipitates had a particular distribution because they tended to form low energy boundaries. Whereas if they didn't have this relationship, the boundaries were presumably more disordered and the crystal distribution was different. You might say, why am I talking about this in this talk? But actually there's two things about it. One is this coincidence site lattice appeared again in a different context about 10 years later. I'll just mention it a bit later. And the other is that it sort of set me up for the next part of my career, which was when I went to the Laboratory of Molecular Biology in Cambridge and was given the task of trying to improve electron microscope images that at that time were of normal bright field images of specimens in negative stain. It seemed quite a daunting task, actually, to improve on what was already there. But I guess one thing about my metallurgy days was that I learned that one saw details in metals quite nicely when you use dark field. And that sort of inspired me to play around with removing some parts of the direct beam from the image. And I began in this way putting a thread across the objective aperture. I used spider's threads because spiders made very small threads, and they of course have a sort of natural adhesive. So the only problem was getting them across the center of the aperture. And the idea was to remove some of the direct beam. What I found was, very surprisingly, was when you had the direct beam in between the thread and the aperture, in this region here, and you had the image in focus, and then you moved the thread into the position, what you saw was a whole range of images being formed which changed with time because the thread got charged up. But if you looked really closely, you saw very sharp images suddenly appeared and then disappeared. This is only for a fraction of a second. And I thought maybe we were onto something good here, and what was happening was that the thread was charging up, and maybe you could control the charge on the thread. And so I learned how to control the charge on the thread by coating it, evaporating gold on it to make it a bit more conductive, and also adjusting the illumination so that you could have more or less secondary electrons scattered from the edge of the aperture to cancel out the charge buildup. And I found out that when you got a few volts in the center of the thread, you got very good contrast images where the phase contrast was uniform from the center of the diffraction pattern all the way out to about 5 angstroms that you could see in carbon films of the images. And this corresponds to a pi/2 phase shift of the scattered wave over this extent, and it was only actually limited by the introduction of spherical aberration term at that high angle. And the images that one formed with this were quite a bit different than the defocused bright field image. This is looking at the tobacco mosaic virus protein, and what the phase shift, and this is quite different than defocusing because you're moving the wave forward rather than retarding the wave like you do in defocus. You could show up the protein in the virus more strongly and diminish the contrast from the stain. And when you did a three-dimensional reconstruction from these faceplate images, they gave a very good representation of the virus subunit structure. I could tell that the representation was very good because the diffraction patterns from the images there corresponded very closely to the diffraction patterns from X-ray structures of the virus that John Finch and Aaron Klug in the lab had taken earlier on. And when I compared them with other structures that people had derived from the virus, other than from ordinary bright field images, the maps were quite different. Whereas we're seeing the subunits, the helical grooves between the subunits there very clearly, and in the other bright field image maps, you didn't see the grooves between the subunits very clearly at all. And so there was a big difference between the structure obtained from this type of image and this type of image. And I realized that the reason that there was a difference there was that this image is emphasizing the protein, whereas the bright field image is emphasizing the distribution of stain around the protein. And as you irradiate the object, the stain distribution changes around the specimen, so it gives you a rather false impression of things. So I learned from that if you're going to get a good image and negative stain at least, you need to use a very low dose. So this got me to thinking about using low doses to get more accurate representations of the structure. But of course, there is the other problem, and that is that the specimen is not well preserved, even in the negative stain, because it gets dehydrated. And one way of getting around that problem that I found was to, instead of using negative stain, was to, this is doing experiments with another specimen, the catalase crystals, was to use glucose instead of stain to immerse the specimen in. The glucose doesn't evaporate in the microscope vacuum and works as a good substitute for water, as you can tell by the electron diffraction patterns, which go out to 3 or 4 angstroms resolution and actually look very much the same as diffraction patterns as I could get from X-rays. Well, at about that time, I was very lucky to be Richard and I started working together. Richard had done some very nice work on X-ray diffraction of purple membrane pellets. Purple membrane contains the crystalline arrays of Bacteria-rhodopsin. And what we did, first of all, was to look at the fading of electron diffraction spots from catalase and from purple membrane. And what we observed was that the spots faded very rapidly. They were extremely sensitive to the electron dose, and after about 1 electron per square angstrom, the spots are sort of were well on the way to fading out completely. And this meant that if we were going to see the structure well, we needed to use a very low dose indeed. We devised a special way of taking the pictures where you have an off-axis viewing screen and a mechanical shutter that moves the condenser aperture in and out of place to open up or close the beam. This is a sort of mechanical version of the electronic thing that they have on the microscope now. And we were able to take photographs of catalase and purple membrane at a very low dose. And the trouble is, in the images, you couldn't see anything, because in the low-dose image, there weren't enough statistics to define the details clearly. And if you took a high-dose image, of course, the specimen was completely demolished. But what you could do, look at the diffraction patterns. We looked at optical diffraction patterns in those days, and we could see spots arising from the crystal structure. So the spots of the signal that become separated in the diffraction pattern from the noise, which is the random distribution of intensities from the random distribution of electrons. And so spatially separates the signal from the noise. And of course, then you know that you can collect the information, you can scan the microscope, collect the information on the spots on the lattice and derive a projection structure. Of course, there are things like you have to correct for the contrast transfer function, which you could measure from the second picture where you've over-radiated the specimen. And you could get more accurate amplitudes by electron diffraction. And so this shows the picture of projection structure of Bacterodopsin after collecting the information from the diffraction spots. And then, of course, we could combine tilted views, get a three-dimensional structure, and finally end up with a model showing the 7-alpha helical structure of Bacterodopsin. Well, I'm sure Richard's going to tell you more about that. That was a brief bit, and of course Richard went on and did some wonderful things getting to near atomic resolution with Bacterodopsin. I sort of went in a different direction at that point, thinking that this was a brilliant way of doing things, and of course the ribosome is the really important structure, and why not do the structure of the ribosome this way? And so I noticed that an Italian scientist, Carlo Taddei, had seen these ribosome crystals on the endoplasmic reticular membranes of the oocytes of hibernating lizards, and I thought these would be an ideal specimen to work on. And you can see the double layers of ribosomes making the crystals, and you can see that they're made up of tetramers when you look on the on-face view. What we found was, which was really interesting, that the double layers, the two sheets of crystals were lying at specific angles relative to each other, and they formed this sort of coincident type lattice. Those were the particular angles that led to coincident site lattice, and this is presumably because the ribosome tetramers like to sit on top of each other, and they sort of jiggle around until they can find a situation where the tetramers are sitting on top of each other. Well, from these sheets, actually, I was able to get a low-resolution structure of the ribosome sitting on the membrane, showing how the large and small subunits are oriented. And I thought this was pretty exciting, and saw this, saw this, they put it on the cover of Nature. The trouble about this structure was that the resolution was very poor. In fact, it was only about 90 angstroms, and it was in stain. And actually, it was left to my then student, Werner Kuhlbrandt, to take the project along further, and he actually looked at it in glucose and gold thioglucose, and did contrast matching experiments at quite a bit higher resolution than this, and worked out the distribution of RNA and protein in the ribosome. Well, Werner then went on and did some marvelous things with the light harvesting complex, and I sort of went sideways again and started looking at nuclear pore complexes. I thought the the nuclear pore complex is a very interesting structure. It controls the flow of molecules, of course, between the cytoplasm and the nucleus. And you could, by adding a little bit of detergent, you could get a crystalline arrays of them dropping out of the nuclear envelope. And the trouble about these arrays was that they weren't at all well ordered, and I ended up, this was work with Ron Milligan, actually doing a three-dimensional structure from a single particle in the array. And we learned then that the nuclear pore was a symmetric structure with big particles actually sitting on the cytoplasmic surface. And one could sort of shake those particles off and look at them, and I thought they looked very much like ribosomes, and I'm pretty excited about this, and showed George Palladie, who discovered the ribosome, and when he visited us once, and George was very skeptical. He said there's lots of big things in the cytoplasm, and no reason to think that those should be ribosomes, although I think it's a very good place for ribosomes to sit, sort of collecting the message as it comes out of the nucleus. But the other thing that George said was that what is the point of looking at the structure out of the membrane? I used Triton to release these pore complexes. You only get a model, not an understanding of how it works. I'm not sure if that's totally right, but it did have a strong influence. I got totally disappointed with this project. And actually I was lecturing at Stanford at the time, teaching neurobiology, and it seemed to me that the thing that you should really work on if you're going to understand how the brain works is the basic unit of communication in the nervous system, which is the synapse. How is the signal communicated from one nerve cell to the next, or the nerve cell to its target? And so I began working on synaptic transmission, in particular the one involving the transmitter acetylcholine. And I'm going to say a little bit more about this now in the last part of my talk. So let me just very briefly say what goes on here, just to remind you. When a nerve cell is activated, it sends an electrical impulse along the nerve cell membrane, and when it gets to the nerve terminal, it causes vesicles containing a neurotransmitter, acetylcholine in our case, to be released, and then bind to channels on the target cell, in our case acetylcholine receptors. The channel's open for about a millisecond or so. The ions flow through. This changes the potential on the target cell membrane, and in the case of the acetylcholine receptor, which is on the muscle cell, the muscle then contracts. So it's a very rapid process, and the questions you want to ask is how can this all happen so quickly? And for me, the interesting part was the postsynaptic membrane, where we felt we needed to answer three questions. One was the structure of the receptor. Obviously, you need to see that to understand how it works. You want to understand the conformational change. How does it open and close? And you also need to know how are the lipids involved in this process. This ion channel, I suspect many others, don't work unless they have the right lipids around them. And this particular channel certainly doesn't work if there's no cholesterol, which is in high concentrations in the postsynaptic membrane, like 1 in 3 molecules will be cholesterol here. So I want to tell you about our progress in these three areas using the postsynaptic membrane that we got from torpedo electric organ. The electric organ is sort of muscle-derived tissue, so what you see there is essentially what you would see in muscle. And by this time, we got past the glucose phase of looking at things, and one was doing cryo-EM because they built good cold stages by then, and of course Jacques Dubochet and his colleagues had invented the technique of rapid freezing to get amorphous ice. And with the torpedo membranes, Alain Brisson, when he was a PhD student, found that they form these long tubular vesicles. And when he came and worked with me, we spent a long time doing initial studies of these tubular vesicles. You can see one going across a hole there in the ice. If you look with a direct detector, they look very, very nice and regular. And if you do some processing, you realize they're made up of dimers of receptors, which form a helical arrangement along the tube axis. And if you compare the arrangement of receptors with the receptors in situ, you find that they both have dimers of receptors, so the packing is more or less the same, and we've done nothing to the lipids, so the lipid distribution is essentially the same as you would see in situ. Well, working with a number of people over many years, we gradually got higher and higher resolution structures from the acetylcholine receptor. First of all, Chikachi Toyoshima made an important development of the helical reconstruction methods using Fourier-Bessel approach so that we could improve the resolution of the structures. Barucham developed a method of breaking the tubes down into short segments and aligning them individually, and so correcting for distortions in the arrangements of the receptors. And then also we worked for a long time with Fujiyoshi, and in particular Atsuo Miyazawa, who's very good at taking images. And this, after a lot of work, enabled us to get a 4 angstrom structure of the receptor. It wasn't a very good structure, and we needed help from a acetylcholine binding protein, but it's in the membrane, and you know, we're confident it was a physiological structure. Now, let me just tell you first, just very briefly, how the molecule is built up and how it works. It's made up from 5 subunits. There's 2 units, 2 subunits with identical sequences, and then 3 others, so the 2 alphas, the beta, the gamma, and the delta. And each subunit is built around a beta sandwich core in the extracellular domain, 4 alpha helices in the membrane part, and a single helix contributing to the intracellular domain. The ligand acetylcholine binds here, and the gate is about 50 angstroms away. And of course, the question is, how does the binding there trigger the gate, opening of the gate so far away? When it turns out that when acetylcholine it binds at the 2 binding sites in the 2 alpha subunits, but this alpha subunit makes the main energetic contribution to the conformational change, and actually pushes against the beta subunit when acetylcholine goes in and the structure sort of closes around it. I'll say a bit more on that in a couple of minutes. Here's a picture of Fujiyoshi, and here's a picture of the microscope with all the films that we didn't use, about 10,000 of them there, because a lot of the images, it wasn't that we had a problem with movement, which all these pictures were taken at liquid helium temperatures, in Yoshi's very special liquid helium cooled microscope, but it was very hard to get homogeneous helical families, and a lot of them were just, the tubes were bent or too fat or something like that. The second part I wanted to mention was the method of capturing the conformational change. And what we did was spray acetylcholine onto the grid and then plunge it into liquid ethane. And we had particles of ferritin in the spray so that you could see where the spray had hit the tubes. And if you made the delay between the spray hitting the tube and the freezing down to, you could make the delay very short by having the spray just above the liquid ethane. We've got, say, 10 milliseconds in this picture, but one can actually do it quite a lot smaller delay than that. And that was quick enough to capture the channel in the open state, because if the acetylcholine interacts with the receptors, if it's above saturating amounts, it is very efficient. 95% of the channels will be open. And as long as you don't get desensitization, which occurs after about 20 milliseconds, they should all be open. And what we found was, or just in summary, that the beta subunit next to the alpha subunit gets pushed in the extracellular domain a little bit. I've got about 2 more minutes. So if you see that it moving, the top part's shifting a bit, and the bottom part tilts outwards to accommodate this shift. And then the part in the membrane, somehow this tilting in the membrane destabilizes the gate, and the helices bend to open. Well, and the final part is we're just beginning to understand the lipids, and I wanted to show you some recent work from using the direct electron detectors, showing something about how the cholesterol is arranged in the membrane. In parts where cholesterol has a very small head group. So in parts where the cholesterol is concentrated, the phospholipid head groups, which are large and strongly electron scattering, are missing. And so you can look in the lipid bilayer, and if you look really closely, there are bits missing beside the receptor. Here's an example there. If you fit the structure to the densities, you can see very, very clearly where these bits are missing. Here's this example next to the blue, which is the delta subunit. Here's another example here. And then if you look at sections parallel to the phospholipid head group region going through the phospholipid head groups, you can see the pattern of where the cholesterol is concentrated. It's concentrated around the edges on one side of each of the subunits, and also concentrated in a patch here, which links the two to a pair of receptors forming the dimer together. Nobody's ever seen a patch of cholesterol in the membrane before, as far as I know, so this part is particularly interesting, and you ask, why is there an extensive patch there? Well, I told you that when the channel opens, the alpha subunit pushes against the beta subunit. And one reason why there's a concentration of cholesterol there is that one wants to rigidify the membrane in that region so that the conformational change can occur productively with the beta subunit tilting out and the delta subunit being sort of anchored in place by the rigid sterile groups of the cholesterol. So we think that the cholesterol there might be play an anchoring role so that the delta subunit can resist the force applied by the alpha gamma subunit tending to push and providing a component to push the beta subunit out. And actually, if the cholesterol is forming a wall there to anchor the delta subunit, then this receptor is also part of the wall, and so you might expect cooperativity between the two receptors. And indeed, when you do single channel measurements, you find that they don't open sort of randomly relative to each other, but open together in synchrony. Well, I haven't got time to say anything more, but let me just summarize. I've told you about my experience in 4 different techniques. I've put an asterisk next to spray freeze trapping because I think this is an ideal technique for single particle methods. It's very easy to see at low magnification where the sprays hit the grid, and you can measure the concentrations from the particles that you have included with the spray, and you can, from the distance out, you can estimate the sort of time of reaction. For us, a big excitement in the future will be that looking at the lipid bilayer more closely, we're beginning to see actually the head groups of the lipids now surrounding. Maybe you can't see it there in that picture, but in the end we should be able to get a complete atomic picture of the postsynaptic membrane. Thank you all for listening to me, and thank you very much to my collaborators. Of course, Richard has always been there for help and advice. And I think I've mentioned the others as we've gone along in the talk: Alain Brisson, Chikashi Toyoshima, Ramin Barucham, Atsuo Mizawa, and of course Fujiyoshi. Thank you very much.
Thank you very much, Nigel, for this fantastic talk. We're a little bit out of time, but maybe really quick, one quick question. If somebody have really quick question. Yeah, so I guess people just would like to discuss it with you. I'm sorry, just because of lack of the time, then we have to carry on.